Nope, I have. Like these are my video cards, and then these are my jazz anatomy cards plus jazz vocabulary. No. No, I do like I put my own. I make my own like. Well, this is what I'm doing, but I would do.
Okay, I guess we'll go ahead and get started. So I don't know if you guys have ever heard of me before or not. I'm Steve Ward. I'm PhD, but I've been involved in clinical departments for most of my career. I was currently the director of the Institute for Biogenesis Research. They already started it, which is focused on reproductive biology. So because of that, we also have become closely aligned with OB/GYN department. So I work with fellows and residents, and just show you that I have some familiarity with the clinic, not a whole lot, but some. So this lecture is supposed to be kind of a review of epigenetics and give you some specific diseases that they wanted to kind of put it together. I'm just given these titles to give some of these lectures, so they don't always make sense to me, but we've tried to make it work. So I'm going to talk a little bit of review of epigenetics right now, which is really a fascinating field. How many of you guys know Lamarck, the difference between Lamarck and Darwin? Do you remember this story? So years ago, a century ago, we were arguing about evolution and how it occurred. And this guy Lamarck said a giraffe has a long neck because one giraffe will stretch his muscles to reach the leaves, and his neck gets longer. And because his neck or her neck gets longer, their babies will have a longer neck because he got a longer neck. The parent got a longer neck during life somehow. That somehow the action of the animals would affect their inheritance. And then Darwin came along and said, no, it's natural selection. The giraffes have a long neck because a baby that was mutated that happened to have a long neck could eat better than the parents, and so that one bred more often, etc., natural selection. And it turns out, so everybody thought Lamarck was crazy, and when I was in school, we all laughed at him and thought he was crazy. But it turns out that there are a lot of things that one can do in living that will affect your genes. And these are inherited. These are called epigenetics. So we do have the ability to change our DNA. This is actually part of our normal function, because every cell in your body has every gene necessary to make your whole body. So when you think about it, the genes to make a brain, which I think is probably the most complicated organ in our body, and the most, because we talk a lot about brain diseases today, and especially by architecture. And I think the brain is the one organ that is most affected by architecture of any other organ. I was trying to think about another one. Everything else can sort of survive. But the brain, you have a little function that changes. Anyway, so we can change our DNA sequences, right? So so the elbow has the genes to make the brain, and this is why cloning works. But the reason why you don't grow eyeballs out of your stomach is because the genes are suppressed, the right genes are expressed. So the genes are modified somehow. And they're modified by methylation, by chromatin structure, by histone acetylation, which we will get to. That's called epigenetic changes. So we can change our genes. We can modify the structure of the proteins that bind to the DNA, and even the DNA itself, so we can modify the expression so that only the right genes get expressed in the right cell types. It turns out that those epigenetic changes can be modified by smoking and alcohol, etc. And so we can change our environment, right? So this gets to be really important. So this is, we're going to give an introduction to genetics. We're supposed to go 2 hours, but I hardly ever go that long because my tea doesn't last that long. So we'll see. There's some things, we're going to talk about DNA methylation, histone codes, and then non-coding RNA. Anybody who's heard me before will know that I have this sort of cheat thing. I want to tell you guys that I think the most important discovery of this century in biology is non-coding RNAs, and that it is coming into your practices. We will show you how it does it. But the bottom line is that we have We have 23,000 genes that produce proteins in our body, and through alternate splicing, that gives about 75,000 different proteins. But we have 1 million non-coding RNAs, and these non-coding RNAs are expressed with the same cellular and temporal organization that proteins are. And these are really important to differentiation, et cetera. And I'm going to give you some examples. Sorry. I won't address any further than this, I promise. Okay, so we'll talk over the basics, and then we'll go a little bit. So let's talk about this. So around the turn of the century, we sequenced the entire genome. So this was 3 billion base pairs times 2 in our body. And when I was in the postdoc, we used to think of 3 billion base pairs as an incredibly large amount of information, because we said we got a textbook, we counted the number of letters in that textbook by pages, et cetera. And in a 700-page textbook, 300-page textbook, it would take 700 pages to make 6 billion, right? And so we used to think about this as an impossibly large amount of information to consider that we could even organize. But now, when we talk about 6 billion, you guys think 6 gigs, and that's not a very big hard drive at all. And that most of us could not conceive of anybody writing a program that is only 3 gigs long that could do everything that a cell does. To take a microscopic egg and just start dividing and become a human being. And so this organization has to be very compact. And there's a lot of things about computer science that I think we could learn about how DNA regulates itself. A lot of this is these non-coding RNAs, by the way. And so when they first started doing this, the whole point of this structure, this is the first organization of chromosome 21 when it was first completely sequenced. The ribosomal RNAs and the centromeres were not sequenced at that point, they are now. But this CG is the methylation sequences. So CPG are the GC sites these are the sites that are methylated. And so they thought it was an important enough thing to look at how it's organized, methylation. So let's look at DNA methylation. So the only sort of chemical modification that we make to DNA is methylating the cytosine. The body uses this to turn on and off genes, and this is one way that it directly functions on the DNA. The problem with it is that when you methylate cytosine, you create a very potential modification, because by putting this methyl group here, it chemically makes this amine group very easily to leave, because the electrons can be transported down here with a carbon. And you can make it turn it into thymine. So it's called a C to T transition. And it turns out that 30% of all cancers are C to T, point mutations in cancers are C to T transitions. And so during evolution, because of this issue, during evolution, most of the CG sequences were evolved out. So the sequence CG itself is very underrepresented in the entire genome, as you would occur by just normal chance. And there are places where it is the normal occurrence by, where you would expect it. These are called CPG islands. And so these are the places where evolution kept it. So there's always this balance in evolution between plasticity and stability. And so we need the stability by not having mutations throughout the body, potential C to D transitions, but we need the plasticity of manipulating our genome and marking which genes we want to express and which we don't. So these are the CPG islands. So the result is that whenever we see what we call a CPG island, cytosine, phosphate, guanine, so it's always CG. That's our sequence that is methylated, the cytosine just before guanine. These are just the places where the CG sequences were kept normally. And these can be methylated. So these are places where proteins will come in and methylate the DNA. And just in general, it's not always true. And there's lots of there's always an exception to the rule. But in general, when the gene is methylated at the beginning of the gene in the CPG island, it's not expressed. And when it is unmethylated, it is expressed. And you can change you can sort of like a rheostat almost. The more methylation, the less expression it is. Okay? So this is in general. So the sequence CG is a palindrome. So whenever you have CG on one side, you have the same sequence on the other side. So that means that methylation usually occurs hemimethylation, right? So when you have a CG site on one strand of the DNA, you have CG on the opposite because the strands are anti-parallel, right? They go this way and then this way. So that's very handy because when you replicate the DNA, if you have a methylated group, there's a methyl group, there's an enzyme called DMT1 that comes in, and every time it finds a hemimethylated sequence, it'll just methylate the other side. So when a cell divides, it can propagate the methylation. So when you go to the trouble of finding a gene that you want to over-methylate, during replication, it's easy just to, it's called maintenance methylation, right? The issue is, though, de novo methylation. This was a big mystery. How does the cell decide to methylate a gene and not methylate another one? Okay? I'm going to give you a hint. It's non-coding RNAs. That the RNA can go and find the gene it wants to methylate, but we'll come back to that later. So de novo methylation is a really important phenomenon because as cells differentiate, they change their methylation patterns so they express different genes, right, during embryonic development. The zygote has one set of methylated patterns. They're different from the paternal and maternal, right? But those have to change as they express into over 300 different cell types as the body develops. And so you can lose methylation simply by diluting it out, right? So if you just turn off the DNA methylase, if you have no DMT anymore, you just replicate it, and as you replicate every time, then you sort of dilute out the methylation. And so most of the genes are no longer methylated. So just by ignoring the methylation, you can lose it, right? So that's one way to lose it. It turns out so that we have a very, very, very strong population genetics program in the Cancer Center. This grant has been going on for almost 30 years. And they've done tons of studies. Hawaii is an amazing place to do clinical studies because of our diversity and because of our compliance of our parents, of our patients. If you end up practicing here in Hawaii, your malpractice insurance would be a lot lower than it would be if it was in New York, where they sue you if you look at them wrong. But here in Hawaii, people work with their physicians very nicely, but they also stay here. It's not easy to drive across the state. And move. It's not easy to move. Once you get in and you're stuck in Hawaii, you like it here, you don't really want to move. There aren't many places like it. So some people can't wait to get off the island, but some people can't live anywhere else. I once had a graduate student that went to Stanford, of all places, and couldn't stand it after 3 years and came back and finished because at UH. Sorry. So we found that Japanese Americans and whites and Native Hawaiians have different responses to smoking and different changes, the racial differences in how their DNA is methylated in response to smoking, for example. But smoking does change our methylating patterns, right? So it may be that mutation is not just causing mutations in the DNA sequence, but it may also be changing the epigenetic changes, right? So we have three types of DNA. The DMT1 is the maintenance methylation that just keeps the methylations going. But DMT3A and DMT3B are de novo methylases. So they work with RNA and other proteins to find the genes that you want to methylate. And then there's MECP2 and other proteins. There are proteins specifically that bind the methylated DNA that help turn it off. There's just one signal. So the major methylation site summary, the major methylation site is the CpG island, C, 5' C, G'. And CpG islands are less prevalent than we expect. So like AT or any other combination of nucleotides, CG is less expressed because of this potential mutation to thymidine. The DNA can be actively, so CpG islands are used to regulate methylation. They're not only at the first part of the gene, but sometimes they're at introns and sometimes they're at the end. DNA methylation can be actively or passively reduced. You can just reduce it by cell differentiation, but there are also specific proteins that will come in and wipe out the methylation actively. And DNA methylation is sort of the first thing that we found that's affected by our environments. This is Lamarck's revenge. So if I smoke, I can change my DNA methylation patterns, and that changes my physiology. And by the way, I can change my methylation in sperm and egg, which can be inherited if it's not cleaned up. So some of these changes can be inherited, these methylation marks. Okay? Let's look at now histone modifications. The histone code was another big surprise, and it's another major discovery of this century that people are looking at. It will come into your practices. People are already devising drugs to fix this, okay? So just to remind you, the histones are these 8 proteins that take this DNA and wind it around to kind of close it. I mean, the problem that the cell has, this topological problem, is that every cell in your body has 6 feet of DNA in your cell. So if you take all 46 chromosomes and you unwind them carefully, you lay them out end to end, it'll be 6 feet of DNA. And that fits into a cell that's 10 micrometers wide. If you scrape the fingernail on the inside of your cheek, you have 10,000 of these cells. So you have 10,000 6-foot pieces of DNA. And so this DNA, it's not a volumetric problem because it's very, very thin. It's like a cable going across the ocean. The ratio of the length to width is huge. So their volume isn't a problem, but the topological organization of it is, right? Kind of like winding a tape in a tape cassette, if you guys know what that is. Anyway, so these histones were part of this thing of the coiling. It's just the first step in coiling this DNA down into it. But they have these histones. There's 8 histones, 4 different, two heteromers, H3, H4, H2AB, and H2A. And there's 2 of these tetramers in the cell. They have these long amino end tails. And these tails can be modified, right? They can be modified by acetylation. They can be modified by methylation and phosphorylation, the 3 major ones, right? So here's what this looks like in 3-dimensional. The DNA wraps around this, and these tails kind of wrap around it. I won't go into the complex of this, but you just have to think the complexity of this is incredible. If you look at all these possibilities, right? So histone H2A has a lysine that can be acetylated and a serine that can be phosphorylated. H2B has 4 lysines that can be acetylated. H3 has a lysine that can be methylated or acetylated, and the methylation can be 1, 2, or 3 on the same amino acid. And when you start to look at this, you see that there's in one histone octamer, which is only every 200 base pairs, there's an infinite number of combinations. I mean, if you start making a code based on the different modifications of these, there's already an infinite modification rate. And most genes are about 10,000 base pairs long, so that's what 500 of these nucleosomes, is that right? 500 of these nucleosomes. And so you start talking about an infinite number of possibility combinations. And these modifications also affect gene expression. Right? And it gets even more complex. So the complexity of our genome is really, really interesting. It turns out that, it's a side story, it turns out that most of what I'm telling you is not going to be on your boards. I'm just trying to actually educate you about this stuff. So I apologize for that part. But I can't change now. I'm too old. I'm 68, so I'm not going to change. So we have 23,000 protein genes in our genome. C. elegans, this 2-millimeter worm that you can barely see with your eyeball, has 19,000 protein genes. Nobody is going to convince me that the complexity of a human being in C. elegans is anywhere near close enough together. What we do have is a genome that is several orders of magnitude larger. Ours is 3 billion base pairs. C. elegans, I think, is like 200 million, something like that. I don't remember the exact number. That extra is the main difference between the 23,000 proteins that we have and the 19,000 proteins that C. elegans has. Is that our extra proteins are all transcription factors. These are genes that will bind and control genes. And the idea that it's emerging, at least from the proteins, not even considering the non-coding RNAs, is that just from the proteins, that C. elegans and human beings are using the same construction materials. But just like you can build a McMansion or you can build a little cottage with the same lumber. It just depends on how complex you want to be. They take the same, building materials, the same set of genes, and just manipulate them differently to create this thing. And one of this complexity is this little thing. It's mind-boggling to me to think about. So we're looking now, H3K9, so the number 9 lysine on H3 can be methylated, right? And so this is from a review. These are called writers, the ones that set up the genetic code, right? And this is a direct quote from this thing. There are several families of histone H3K9 methyltransferases. So what they're saying is that they've identified several genes, several families of genes that modify this one lysine on this one histone. And the implication is that this may be, there may be a similar complexity to all of these different modifications. And these different methyltransferases can be expressed in different cell types and go to different genes depending on what cell type it is. And we add this complexity to all of this, that probably these things are driven by RNA that directs the histone methylase to the right gene. Then we can see that there's a huge complexity in how we organize our genome. But what I'm going to hope to show you towards the end is that people are starting to look at this as treatments. Okay? And this will come into your clinic at some point. So there's histone writers, and then there's histone code readers. So once you methylate this lysine, there are proteins that bind to it that will either turn off the gene or turn it on. That's all you need to know. So there's writers, the enzymes that change the histone pattern, and there's writers and readers. So this HDAC right here is a histone deacetylase, HDAC. That's a very interesting protein. There are several inhibitors of different HDACs that people are using, I think we'll tell you about later on for Parkinson's disease, because this protein can modify how the epigenetic code, right? This HDAC comes in and it takes off the acetyl group of lysine. So if you methylate your lysine, sometimes it brings in this protein, which deacetylates other things, which changes the epigenetic signal. We're just now beginning to get this complexity. And I will tell you that when I was maybe 35 years ago, I was a young assistant professor. I used to be young once. And I was asked to give a talk to a group of urologists who were just now panicking because PCR was coming into their clinics and they had no idea what PCR was. You guys know what PCR is, right? Everybody? PCR is amplifying genetic, so we can amplify DNA. Most of you have probably done it volunteering in labs, right? So you just amplify DNA. But this is now one of the main techniques. This is how I can take my kid to the pediatrician with a stuffy nose and we decide, do we give him antibiotics or not? We give him antibiotics if it's bacterial, but we don't if it's viral because that's a waste. So they take a swab. And later that day, they tell me if it's viral or bacterial because they do PCR. This used to take days of culture. But this is a very simple test. So this is why I'm telling you all this, because this stuff will come to your clinic later on. This one slide is the best image of the histone code that I've ever seen. So they took these ES cells in culture, and you can differentiate ES cells into neuron progenitor cells or murine embryonic fibroblasts. So this is fibroblasts versus neurons. You take these ES cells and you differentiate them. You look at, we can map now using complicated genomics, et cetera, but we can map now the histone modifications on every gene by pulling down the histones and looking at them. And so what we look at is, here's the histone H3, and I urge you to kind of look at this and understand this slide. If you understand this slide, you understand the histone code. So here's two different modifications. There's 3 methyl groups on H3K4, and there's 3 methyl groups on the same histone but K27, right? So in the ES cells, the H3 is methylated, the K4 is methylated, but the K27 is not. When it differentiates into neurons and murine cells, this methylation pattern holds in this gene. When you look at a different gene, the pattern is different, and the pattern splits when it differentiates. And you can find just about every combination here in these different genes. And remember, there's 23,000 genes. And this is just one step in differentiation. So normal differentiation, and these guys did this in culture, but they verified it in tissue of adult mouse cells. So in just one change of an ES cell, differentiating either to neuron progenitor cells or fibroblast progenitor cells, you get all of these changes throughout the genome in this epigenetic charge, right? So it's not a great leap of faith to think that these epigenetic changes would be very involved in disease, right? Why do different people with diabetes respond differently to different treatments? It may be related to these epigenetic changes, okay? Again, our epigenetics powerhouse in the Cancer Center looked at some of these epigenetic changes, and they found differences in race, in smoking and alcohol drinking, etc. I'll just run this by you very quickly because it's not related to epigenetics really, but there are, we can change the histones by post-translationally modifying them, or we can also replace the histones with slightly different variants. So histone H2A is the main variant in most of the histone complexes, but there's a few other genes that sometimes the cell can insert into it. And there's slightly different amino acid sequences, and they have effect. And I found this one paper years ago that said that memory might be affected by different histone variants that are put in the histones in ways that we don't recognize, probably due to some epigenetic changes that occur in that histone that changes the expression of the neuron, right? Developing the neuron, changing the ways that we don't understand. But our differentiation of different cell types and in different disease states changes these epigenetic patterns in the chromatin, which changes the expression patterns of cells in the DNA. So, summary of the histone code. The histones are the primary component of chromatin, like DNA plus protein. Histones can be modified by methylation, acetylation, or phosphorylation. The histone code is constructed by writers and executed by readers. Makes sense, right? Histone modifications differ in different genes and different cell types. Right? This is the most important part we have, and you can add to this in different disease states. They can be altered by the environment. So by smoking, by drinking, by exercise, we can change our histone modifications, because all of these changes in our body have effects down to the metabolism, down to the molecular level, and to our genetic level, right? And the histone modifications can be altered by environmental effects, so smoking, etc. I just said that. And there are different variants of histones, okay? Am I going too fast? Is this okay? Is this boring you? Okay, let's look at non-coding RNA. So this is my personal sort of, I feel like, for a while, when I was a kid, I was really, really into religion. And so I feel like I'm proselytizing on RNAs, but I want to tell you a story that's old. It's 1990s, before you guys were born. And there was a woman at Stanford, Helen Blau, very, very well-known scientist. This is one of the coolest papers I ever read in my life. And she was trying to find the gene, to identify what is the gene that caused myofibroblasts to go to the next stage. You could do this in culture. So this was a differentiation phenomenon she could study in culture. She was trying to understand differentiation. And she did this really amazing, assay that she had to develop first in order to find the gene. And she found the gene, it's troponin C, which makes a lot of sense because troponin C is part of the protein. That is one of the proteins that's in the muscle cell, so that's great. She says, I've got it. And then she did one control too many. She took, in an RNA, there's the coding sequence which actually produces the protein. There's the 5' UTR, which is the untranslated region, which controls, kind of how you express it. And then most genes have this very long 3' UTR that's usually even longer than the coding sequence. And that also helps with when you express it and stuff. And so she transformed cells with just the 3' UTR, right? For the full length, you got about 20 things done. So this is all the things. Then she took the 5' and controlled, no changes. It was really nice. But when she took the 3' UTR, she got a better differentiation than when she had the whole gene. And she did this over and over and over. So we're talking about a component of the mRNA that doesn't make a protein, and that caused the differentiation of the cell type. This was the first paper that I know of that really focused on how non-coding RNA can direct differentiation, right? And so now people are really looking at this. So now, in the last 25 years, since they've sequenced the first part of the human genome, they've done lots and lots of studies, and these sort of studies are becoming petering out. So in 2005, about 5 years after they released the entire sequence, somebody said, well, what percentage of chromosome 10 is expressed in cell types? Because we know that only 2.3% of the entire genome is coding. Only 2.3% makes amino acid and proteins. The rest of it, in our great wisdom, we used to think 40 years ago that it was just garbage DNA. We used to call it garbage RNA, or that we thought that this was a necessary byproduct of evolution trying to be so complex that it had to be messy. In fact, the complete opposite is probably correct. That this evolutionary history of our genome, which is about 4 billion base pairs, 4 billion years, is probably honed so well because it's a life or death thing that every gene, every sequence there probably has a function. And they found out that 80% of the chromosome was produced RNA, right? And so if we looked at this, it says there was, the intronic DNA was about 26%. Sorry, ESTs and mRNA were about 17% total. So these were about, ESTs are express sequence tags. Those are genes that we don't know if they're up, but RNAs for sure we recognize. So only about 17% of this 80% was RNA that produced coding regions. The rest were intronic and even intragenic. So there's 31% of the transcripts are in between genes, not even part of the introns or part of the non-coding sequences. So the implication is that we used to think that this was just an accident of a messy program for getting the coding sequences that we wanted. That's the way we actually looked at this. Trash, garbage DNA, that's what we call it. But we're now starting to look at that every one of this 31% has a specific function, right? Now, I don't want you to memorize or even understand this slide. I do want to show you, just walk you through the complexity of how non-coding RNAs not only turn off transcription, but close down the chromatin so that it stays off, okay? So we start with these siRNAs. These are small, short, inhibitory siRNAs. Somehow we knew that this double-stranded RNA would turn off a gene. It binds to a protein called dicer, which cuts it up into much smaller sequences. This complex binds these double sequences. This is a complex of proteins. It makes it single-stranded. So now it's single-stranded. So now this turns out to be a guide for finding an RNA that's being expressed. This RNA can hybridize to the RNA that's being expressed, but it also brings all of these proteins to the chromatin structure. Those proteins start. Now, the first thing they do is make a lot of more double-stranded RNA, which goes into the cycle. But they also bring in proteins that acetylate the proteins, that acetylate the histones, and then methylate the DNA. And so that closes the chromatin down, so it's no longer transcribed. What's beautiful about this system is that these same proteins, this Argo and Dicer, all of these proteins here, they don't care what the sequence is. They will take the sequence and say, okay, here's handing out your papers. Here's your instructions for which gene to go out. Here's the RNA. And then whatever gene matches this RNA, you do your thing, right? So we have this very complex system, and it's manipulated by small RNAs, which tell it which genes to go to. Now, immediately you can think of these small RNAs could be targets for therapeutic changes, right? If I'm overexpressing or underexpressing, I can use the same thing to open up a gene as to close it, right? And these are becoming therapeutic targets. These are called microRNAs, and people are starting to use these as therapeutic targets, right? In your practices, you really need to understand the drugs that you're giving to patients, right? It's really important. You have to understand, we all know what antibiotics do. They inhibit, transcription, translation of protein genes where they don't inhibit us, but that also affects our gut microbiota. So we have to, by understanding what's happening, right? The more you understand about the drugs and the treatments you give, the more control you have over it, rather than just being at the mercy of the drug reps that come into your lab and say, we've got this wonder drug and use it. So non-coding RNAs make up the large majority of RNA that's produced in cells. This RNA controls gene development, gene expression, and it's a very important function in disease. Non-coding RNAs regulate methylation and histone modifications. And we are just now beginning to understand this. By the time you guys get through your residencies, we'll have another huge wave of, this knowledge of non-coding RNAs is expanding exponentially, right? And it may be by the time you guys retire that we have changed all of biology to focus on these non-coding RNAs other than proteins, right? The cool thing about RNAs, non-coding RNAs, is that through PCR and DNA sequencing, we can make probes we can amplify it very easily. We don't need antibodies. All we need is the sequence. We plug it into a computer, I make primers, and I can amplify it tomorrow, right? Understanding and analyzing non-coding RNAs is a lot easier than understanding and analyzing proteins. And so because, more than most of the genes are non-coding RNAs, this opens up these things to treatments. So here's just an example of how they figured out that these non-coding RNAs these small inhibitory RNAs can be used to bind directly to the RNA to inhibit it, to make the mRNA stop translating it. But it can also come back to the chromatin like I showed you already and close down the expression and turn it off permanently, right? And these non-coding RNAs will direct histone methylation, which then directs DNA methylation, and then this propagates for a long time, closing down the entire gene, right? So these non-coding RNAs are really directing a lot of these transcriptional proteins to the right genes, right? So it makes a lot of sense that you can control gene expression through RNA. Okay, so I will admit that the first part of this lecture I'm much better at than the next part, but we'll try to go through this next part as we go. So let's look at epigenetics and diseases. So again, we have to think about these. People have started to look at how so the idea is that if I can change my epigenetics, methylation in particular, by smoking, by drinking, by overeating, by exercise, by changing my habits patterns, by stopping drinking, by stopping smoking, does that affect my sperm and my eggs? Right? And it turns out it does. And so this idea took a long time for people to really get. But there are people, one guy in particular with sperm has been looking at this for many years. And people are starting to understand it. There was a study in Australia many years ago where they showed that if they overfed a rat and they made it, a male rat, obese, mated it to a normal weight mother, that the progeny had a greater tendency to be obese, right? And so we can promulgate these things not only by habits, by teaching our children habits, whether healthy or unhealthy, but we can also promulgate them through our genetics or epigenetics or DNA. And the thing about pregnant mothers is that this is a picture that you show in OB/GYN clinics now. That by smoking, a woman affects 3 generations, right? Because women are unique in the sense that males produce sperm they produce the germ cells after they're born and after they reach maturity. But females produce oocytes in their bodies when they're still in the fetus. So if a woman smokes while she's pregnant with a girl, she affects her own body, changing the epigenetics, but she also changes the epigenetics in her child, her fetus, but also in the eggs of that fetus. So 3 generations are affected by this smoking. It reminds me of a thing in Proverbs where it says, the sins of the father will be visited upon the sons for 7 generations. And I used to think this was just that attitudes are hard to die, but genetics can also change, right? Same thing is true for sperm in eggs. We do change our sperm at least in 1 generation. So obesity, smoking, and alcohol will affect a man's sperm. Age affects a man's sperm. It's not true that the older a man gets, he can always have healthy children. It does change. And so we can change the epigenetics of our sperm by our attitudes. And the good news is we can change them back, right? So if we have a healthy lifestyle, if we go back to a healthy lifestyle, if we alter what we're doing, we can change it, right? So, like I said at the beginning, there's a lot, most of the diseases I'm going to talk to you about now are neuronal and in the brain. And I was thinking about that this morning, that I do believe that the brain is probably the organ in the body that is most affected by cellular architecture. Right? The liver, you can take out 3/4 of it in a rat, and it will grow back to its normal size. And it's mostly the same thing over and over again. The kidney also has some major architectural things, but if you cut it in half, some of it will still survive. The colon, people have these things called tortuous colons that they're not fun to live with, but you can live with it, right? Many things in our body we can change. Muscles grow bigger or smaller, but the brain has this very specific architecture that once you start killing just a few cells in one nucleus, you change behavior. And I think that's why there's so many of these diseases that I will talk about are affecting in the brain. So neurons are non-dividing in most cases, but DNA methylation and its regulation controls gene expression. So in Alzheimer's disease, right, there's an overall reduction in methylation, but there's also a gene called REST that is through the recruitment of HDAC, which is this epigenetic modifier, the one that takes off acetylases from histones, can change gene expression. RET is a loss of is a mutation of MECP2, so that's one of the readers. So this is a protein that binds to methylated DNA to turn it off, and the mutation in this gene causes Rett syndrome because a lot of these genes are overexpressed, and it may be caused by aberrations in DMT3A, which is de novo methylase also. And there's something called Tatten-Brown syndrome, where DMT3A, so remember, this is the gene that causes de novo. This is the protein that methylates DNA de novo in response to all these things, which can cause something like autism spectrum disorders and other stuff like that. So there's lots of genes, of proteins that change. Let's look at Parkinson's for a little bit. So Parkinson's is a loss of dopaminergic neurons. I told you I'm not very good at this. In the substantia nigra, because these are actually death of neurons, right? And these are death in neurons because of the accumulation of these bodies called Levy bodies that are, these are basically proteins that start to accumulate alpha-synuclein. This is a common theme in neuronic disorders where you have a protein, a structural protein that just accumulates, and it causes these huge things and kills the cells. Patients have all these problems with muscle rigidity and stability, etc. It can extend to non-motor functions, including depression, etc. What's interesting is that there's only a 10 to 15% familial disease. So that suggests that this is new onset. It's epigenetic. It's a non-genetic cause is what this causes, right? And the late onset of Parkinson's is the one neurological disease that gives us some hope for intervention. So it turns out that there is a lot of epigenetic components to this disease. this protein, so there's this protein SCNA, so SNCA, that is normally expressed, but when it gets overly expressed, it causes these fibrils to form, which kills the cells. And this overexpression is related to overacetylation, deacetylation, causing overexpression of the gene. So there's a decrease in methyltransferase binding, etc. So there's a relationship between the decrease in methylation, the increase in the expression of the gene, and the fibers being caused. So if we could change that methylation pattern, we might be able to get through it. So if we look at the epigenetics of Parkinson's disease, right? So here's this protein, alpha-syn. So this protein is overexpressed. You normally need it, right? But if it gets overexpressed, it causes these conglomerates that kill the cells. There's a protein called FGF20 that will inhibit the production of this protein, okay? But some of these proteins, the mRNA has a mutation that doesn't bind to this small RNA, MIR43. So this is a microscopic RNA. It's a microscopic. This is a small RNA, about 200 base pairs. It is one of these million genes that I talked about that is normally expressed. This hybridization of this small non-coding RNA normally controls the expression of this protein. So this small RNA will bind to the mRNA by hybridization and cause the inhibition of the production of this protein, which regulates the syn. So basically, this small RNA regulates the amount of alpha-syn that you produce. So if there's a mutation in this gene, which doesn't allow this to bind, or if there's a mutation in this that doesn't allow this to bind, it doesn't allow it to bind to this, the alpha-syn gets overexpressed and you get this gene. Another one, miRNA6, binds to HSP70. There's a whole group of proteins called heat shock proteins, which we now know function to help proteins folding and keeps them separate. So HSP70 really helps in preventing these proteins from aggregating, because this is a protein that helps the structure of the protein form normally. But there's a small RNA which inhibits this HAP, which normally inhibits the aggregation. So if this is overexpressed, then it increases aggregation, right? So this one normally, this one inhibits. So if this is overexpressed, if this small RNA is overexpressed, that inhibits the HSP70, which allows this to aggregate too. So this SCNA, this alpha SN gene, this alpha N protein, its first intron is hypomethylated, so that's a decreased methylation. So this causes upregulation in the production of the protein. So that's one possibility. And then there's these other small RNAs that are affected in this as well. So this is an example where demethylation causes upregulation of a protein, which causes the aggregation of these bodies in the cell, which kills it. So isolated, and so they started to look at this, and isolated neurons in Parkinson's disease patients did have increased histone acetylation and decreased HDACs, which may increase the demethylation of the protein. And several mRNAs are also being found to be associated with it. So we don't really understand what's going on yet, but it looks like Parkinson's disease is one of these things that's expressed, that's caused by epigenetic changes, and these epigenetic changes are expressed by small RNAs. So where is therapy in all of this right now? It turns out that there's a weird thing that we don't really understand, that epigenetic changes in the brain can be mirrored in circulating blood cells. And several different studies have looked at this and shown that when they were able to isolate the tissues and look at the blood of the patient, they could see that the epigenetic changes that were modified in the brain can be affected in the blood. Somebody actually here was looking at this as well. And so that would be really cool if that were true. You could just take a blood sample and diagnose what are the methylation changes that the patient is going through in their brain. I don't really know how that works. But the hope is that if a gene is epigenetically modified, if a disease is caused by epigenetic changes as opposed to a mutation that the patient has, then we can then go and control it, right? You can't change the mutation in every cell type, it just doesn't work. But we can certainly deliver small RNAs to change the expression patterns of the genes. So this sort of gives us hope that we may be getting close to finding a cure or treatment for Parkinson's disease at the molecular level. So one epigenetic therapy is modifying these histone code that I was telling you about. Right? So remember I told you that the histone code could be acetylation of the tails. And HDAC is a deacetylation. So if I have a gene that's caused by over-acetylation of histone on a gene, then I might be able to treat the patient, right? And again, remember I said that there are several families of these H3K9 methyltransferases. There are also several families of the HDAC. So HDAC you can think of as an epigenetic eraser, sort of a writer. It takes away the acetylases. So there's several families of these HDACs. And so maybe by identifying specific HDACs for a specific disease, we can hone in on treating the disease. And so people are looking at this now. So several drug companies are developing these epigenetic therapies. And the main goal is to develop more specific HDAC inhibitors to reduce side effects. Because if you're isolating a specific HDAC, if you want to inhibit the right HDAC variant so that you only affect the disease states and not overall, right? Kind of like the antibiotics and the microbiotics. HDAC overexpression can reduce memory and synapse formation in mice. So that maybe an HDAC2, for example, has been increased in PD patients, just one study. So there's a way that maybe if we can find inhibitors to these writers and changing the histone modifications, maybe we can affect Parkinson's disease. This is from a review that's almost 10 years old now, but there are a lot of different pharmaceutical companies now that are working on this. And they're working on specific inhibitors to specific HDACs. So we're talking about drugs that will change the epigenetic modifications of chromatin that express expression, right? That's all you need to see from this slide, okay? And there are different phases of development or work. So we have these three small RNAs for Parkinson's disease that are potential therapies for targeting drugs as well. So not only looking at chemicals that inhibit the enzymes that modify the histones, but once we identify an mRNA, an miRNA that is directly responsible for directly responsible for a disease, that's easy to attack because we can develop RNAs that will hybridize to its stable RNA-like structures that will find these sequences and kill them and hybridize them and kill them off. And we can change that very easily. It's just a matter of doing it. This is what we did with the RNA vaccines for COVID, right? We just injected a bunch of RNA. And that RNA was taken up by cells and it made up the proteins and it raised antibodies, right? It was very simple to do. So this idea of using RNAs as a drug is very possible. And we have the technology to do it. We're really now only waiting for the right sequences that we know to attack. Once we have an RNA that we know we want to kill that won't hurt you as a side effect, then we can start treating patients with this. So any disease we have that's related to an RNA, we can attack. So epigenetics is still a future goal, but again, I will argue that by the time you finish your residencies and maybe fellowships, there'll be more of these drugs on the market than there are right now. there's a lot of pharmaceutical companies that are working on this. Now, the thing about pharmaceutical companies is they don't like to waste money. So if they don't believe that this is gonna, if they don't have evidence that this is gonna really drive it forward, they won't waste their time on it. But if they do have evidence, they will go after it. Again, I'll give you the COVID vaccines as an example. You'll have to wait for a lot more, but we're starting to get the knowledge going. Okay, so this last section is, there's a bunch of diseases that are related to triplet repeats or other repeats. And I really have a hard time fitting this part into the lecture, but I was asked to do it, so we'll do it, and we'll do it anyway. So there's a lot of sequences that are highly repeated that once you get replicated, that somehow you get this, during replication, you can get caught in a cycle and you can amplify it. So there's lots of diseases that are related to this. This is a really nice picture from a review that I looked at these triplet diseases. So you're talking about repeats, usually CAG, but other sequences are working on. You can amplify them. And depending on which part of the gene it's amplified in, it has a different effect. If it's amplified in the 5' UTR, it will have an effect usually on the control of gene expression by methylation, mostly. If it's in the intron, again, same sort of thing. If it's in the coding region, now you're changing the sequence of the protein itself. You're changing the structure of the protein. You're not changing how often it's expressed, but you're changing the sequence of it. And in these non-coding regions also, you're sort of controlling the control of the expression of the normal gene. So there's sort of two really types of changes, right? You can change how the gene is transcribed or whether it's produced, or you can change the protein itself through the same type of mutation, right, depending on where it is. So here's a list of the diseases where you can see different places that occur. So these are all these different diseases, these very difficult, most of these are incredibly debilitating, and a lot of them are fatal. But these are just the repeats that are happening. Only a few of them are in, well, a lot of them, some of them are, about half of them are in exons, but a lot of them are in introns, the 5' UTR or the 3' UTR. So these are all either epigenetic, these three are all epigenetic. This is sort of epigenetic, it's in the altered RNA, but then there's some that are altered proteins as well. But it's the same mechanism genetically, just the amplification of these triplet repeats. Okay? In general, these repeat expansions cause neurological diseases, right? So at least 10 different diseases have CAG repeat. CGG is a Fragile X syndrome, and that's also changed. We'll go through some of these individually. While a majority of them are triplet, there are some documented cases with more than 3. So there's a repeat with CCTG, for example, and other cases. So there are other cases. It's not always just a triplet. In general, these things, some of these features are that these arise from sequences that are already repeated. In other words, you need a certain repeat for the gene to function, whether it's expression or it's the structure of the protein itself. These expansions are unstable, so they're sort of dynamic. Once they start happening, they can increase, and they can increase with generations, right? So you can sort of, they have this anticipation I'll talk about a little bit later. In general, the longer the repeats, the more severe the disease. So you can have different stages of the disease depending on what the functions are. So Fragile X syndrome, for example, if a father or mother has ovarian syndrome, you know, not really debilitating, but it can cause some infertility, then that's an anticipatory diagnosis for the son or daughter having Fragile X syndrome. Okay, and it's a highly variable phenotype, reflecting on where it is and what's happening. So the factors that are contributing are the sequence of the repeat, the size of the repeat, the location of the repeat within the gene, whether it's encoding RNA or protein, the function of the gene, can you live without it, does it really hurt, and the extent of somatic instability. So there's two sort of major things going on with these diseases. One is a toxic gain of function. It causes a problem. You know, you have a bunch of proteins that are produced and now they aggregate, killing the cell, or you have it overexpressed for some reason, or a loss of function, where you turn off the gene and it's not expressed well enough. Right? So let's look at a couple of these examples. And so this gene, this example, SCA, there's a whole group of cells of spinocerebellar ataxia type 3. These are all caused by changes in the coding sequence of the protein. So the protein structure changes. So when you have an amplified gene sequence, more normally you have a lot more of a group of them. Let's say you have a string of glutamines that you need about 10 to function, but if it gets to 20 or 30 or 40, then it can be devastating. So something that you need that you can increase. And in this case, this is a CAG repeat. It normally produces glutamine, I think. 12 to 40 is the normal repeat. So you can have anywhere between 12 and 40, the protein functions just fine. It needs at least 12 to 40 to function. When you start amplifying that just a little bit, when you get to 50, you have sort of intermediate repeats. By 60, you get some weaknesses. By 70, you have ataxia, and then you get complete dystonia later on. So increasing the number of glutamines in this protein causes these problems. And these are several different types of spinocerebellar ataxia type 3. And it basically increases with the number of repeats that you have. Okay? They all increase with the number of repeats you have. This is just a list of these diseases that happens to all of these and what these proteins are. So these proteins can have different functions. So this is, in one case, it can be a scaffold protein. So this is a structural protein inside the cell. It can be a hormone-dependent transcription factor. It can be another code. So the transcription factor would then cause overexpression of something else. Or it would inhibit the expression of something else. So there's lots of different proteins that can be affected. Let's look at Fragile X now for a little bit. So this is an example of a protein. The gene is highly repeated, but it's the 5' UTR. It's not the protein itself that's affected. We're talking about the gene, the part of the DNA that usually causes the regulation of the protein. Right? And so Fragile X is actually a devastating syndrome. Right? It's the most common cause of inherited retardation in 1 out of 4,000 males. It also causes ovarian dysfunction, tremor. It's X-linked, so that means that males are much more are susceptible to this than females because there's only one X chromosome in the males. And it's named for the bent karyotypes in the X chromosomes. And this one thing really fascinates me. If you look at a picture of an X chromosome, of an actual X chromosome, and this is something you can see in the microscope, the X chromosome looks really different because of this repeated thing. And this change in the DNA sequence alters the chromatin structure. I still cannot really understand how changing the repeat from 20 to 200 will make this big of a change in a chromosome that is 240 million base pairs long. Right? There's a lot of things going on here, but it does tell you how the DNA sequence can affect the chromatin structure. Right? Somehow this chromatin is not closing up and not folding the way it should to make a mitotic chromosome just for the separation. This is how people saw it at the very beginning. So just going from 50 to 200 changes the structure of the mitotic chromosome. So must be some signals with proteins that are binding it, etc. In this case, you get a protein FMRP that is synthesized in normal. But in the Fragile X, this protein is completely absent. So the structure of the non-coding part of the gene causes changes in expression of the protein. If you could make the protein, it would be normal, right? So the protein that is affected is a transcription factor. So that means it affects the expression of many other genes, which makes sense now because you have this amplified effect. You take one protein, you change it. It's not just that this is not a disease where it's causing an aggregation or lack of aggregation, but you're closing off a transcription factor. And during development, which affects brain development, that also makes sense. So it's an amplified thing developmentally. We don't really know exactly which genes this transcription factor activates, but it probably activates several genes which activate several other genes, and is probably expressed differently in different cell types. And the same transcription factor can affect different genes in different cell types as well. So it's probably a very complicated sort of thing going on. So, and this is what I meant by, you may see this in your clinics, right? You can anticipate this sometimes. So normal 44 repeats with an interruption every 9 to 10 repeats. So this is how it works. See, I repeated my E's here in this word, so sorry about that. Pre-mutations, okay? So 45 to 200 CG reads, you have reduced expression of this transcription factor. So it's not really strong, but you have a reduced expression. And this is associated with primary ovary insufficiency. We joke a lot in urology about how many genes are affected, both the brain and the testes, because, man's controlled by it. But it's also true that there's a lot of genes that are expressed in the brain and the ovary as well. It just tells you how many times the cell uses the genome for different things, right? And so when you have a woman with primary ovary insufficiency, you want to know if she has this mutation, because her oocytes, when she's replicating the DNA, might have even more repeats, which could lead to, her bearing a child that's mentally retarded, right? So this is anticipation. It's interesting that, this is a mutation that you see in the ovary, but when it gets worse, it affects the brain, right? There was at least one documented change that went from 59 to 538 repeats in one generation. So father to son or mother to daughter, I don't know which. So this does, so it can happen during some replication, right? So the large CG content makes it difficult to sequence. However, that's not so true anymore. I just found out through a grant that I'm writing that we have the ability right over in the BSB building, a guy named Yuping Deng, I don't know if we'll hear from him or not, but he's a world expert in DNA sequencing. So he has this thing called a long sequencer where they sequence genes directly in a line. So that's how people are now seeing these highly repeats. So we can do this better now. And that's number 2. And so it's important for advising patients with pre-mutation. So again, let's go back to this normal thing of generally, if you have the CPG island in front of a gene, if it's methylated, you turn off the gene. If it's not methylated, you turn it on. And what we're really talking about is in Fragile X, you're amplifying the CPG island so that it's much, much longer. So the chances that it'll be methylated will be much stronger. And the signal that happens when it is methylated is also much stronger, right? And okay, so the Fragile X sequence, the CGG repeat, has many CPG sequences that can be methylated. When you increase this level of methylation, you have a much larger signal for turning off the gene. You turn off this transcription factor during embryonic development that can relate in huge, devastating neurological diseases. But you can see it in the previous generation, at least in women, possibly in this ovarian syndrome. So the CG repeat in the mRNA can hybridize to CG repeat in the gene. So what happens is that when you have So the longer the repeat, the more likely it is the mRNA will bind. So you have this mRNA itself, right? And this mRNA can come back and hybridize to the gene itself. And because this is a highly repeated sequence, it's easy to find it and turn it off. And it can also go through this sort of thing structure. It makes a really easy target to turn this off as well. So the RNA itself, by expressing the gene, can come back and turn the gene off because you have a lot more signal now that will find this thing and turn it off, right? So this hybridization can make it shut down. So this is one example, a patient with a full mutation inherited from mother who got smaller but still expanded repeat from her father. This patient develops full Fragile X syndrome with mental retardation during development. The patient has neurotoxicity because of the epigenetic downregulation of the gene, as we discussed. The maternal grandfather developed late onset Fragile X tremor. So this was an anticipation that you might think that the mother or father would have had, right? No, the mother. The same mutation is differing only by the number of repeats, increases the transcription when the number is lower, but decreases the transcription when it's higher. So this is just a slide about anticipation. It's not really it. So Fragile X syndrome is caused by the amplification of the CGG repeats in the 5' UTR of the gene, so the beginning non-coding part of the gene. This amplification results in an aberrant epigenetic regulation of the gene. So you have increased methylation, you have increased RNA, non-coding RNA binding to it, turning it off, and closing the gene down. The regulation is both by DNA methylation and non-coding RNAs, but as I've showed you, these are two very related sequences. If we knew about this, you can maybe think about treatments that you would give a pregnant mother for a lot of non-coding RNA to bind up the other part to prevent it from happening. So then the last one we'll talk about, I think, is Huntington's disease. Is an inherited disease that causes the progression of the breakdown of the nerve cells. So this is another gene that's expressed now. We're coming back to the coding region. So this Huntington's disease is caused by an amplification of a DNA sequence that increases the gene expression. So most people have this normal gene with 30, so most people with Huntington's disease develop signs and symptoms in their in their 30s or 40s. But it could emerge earlier in life. And when it does, like before the age of 20, it's called juvenile Huntington's disease. And this is much more severe and usually ends in death very early. Medications are available to help manage the symptoms, but it can't prevent the physical and mental behavior decline associated with the clinic. Okay? There's a loss of neurons. Okay, so when you lose neurons, it's really hard to get them back. About 57% of the cells are lost in parts and 67% in another part. Other neurons are also lost. There's about 300 grams of brain tissue that was lost in some patients when they looked at them posthumously. The protein that's affected is more than 3,000 residues long. It normally has 16 to 20 repeats, resulting in a small polyglutamine tract, which it needs to work. The mutation has more than 35 repeats. So it doesn't take long to kill this gene, to kill this protein, to make it toxically non-productive. The larger the repeat, the earlier the onset. Okay, so it's easier to kill the cells. Most adult onset cases have about 40 to 50. So the mutated protein has more than 35 repeats that tends to aggregate. So this is another one of these proteins that has huge glutamate proteins and it tends to aggregate. And this dysregulation causes a myriad of problems, including mitochondrial changes and sort of global effects of translation. But there is some potential therapy, right? So heat shock proteins, we talked about before, have the ability to disaggregate proteins that are congruent together. So in culture, they've been shown to dysregulate them. Antibodies have also been shown to reduce cell death. So antibodies to caspases have been shown to reduce the cell death. Caspases might degrade mutant proteins by producing toxic hunting fibrils that would aggregate more. And there's antagonists, right? So caspase antagonists, these proteins that inhibit. So the problem is the caspases that try to degrade the protein cut up pieces that actually aggregate even more, so you can inhibit the caspases to prevent the aggregation. That's one potential therapy, right? So we can also use HDAC inhibitors to decrease the expression of these proteins slowly. And siRNAs can also be used to decrease the expression of this protein as well. So one last one is myotonic dystrophy. This one is also epigenetic in the 3' UTR, right? So this gene is caused by an expression of CTG repeat in the 3' region of this gene. The DMK RNA, so the RNA for this protein, binds and sequesters different proteins preventing function. Now remember when I told you about troponin C, and the 3' UTR was affected, Lau found that the 3' UTR caused this differentiation of pre-muscle cells to the next stage of muscle cells. In this case, we have an RNA gene, the protein. The protein doesn't really make a difference, but it's the 3' RNA. This 3' RNA binds to a bunch of proteins to help regulate transcription, etc. If you expand the 3' RNA, then that sequesters a lot of these proteins more than it should, and that causes the problems. So it's really sort of a complicated issue. It's the 3' UTR, not the protein that's being transcribed, that's sequestering other proteins that regulates the cell itself. Right? And so again, I reminded you of that. So this is just a picture of that. So you have this, normally this gene, the RNA, will bind to these proteins and sort of sequesters them and keeps them from doing other things in a normal pattern, right? It regulates the normal way. But when this part is amplified, these proteins are really now sequestered and they can't even get out of the cell to do what they need to do, and that causes the protein. A therapy, an obvious therapy, might be to add a bunch of small RNAs that will either bind to these proteins and just go out, right? Or bind to or treat with RNAs that will treat these proteins that will cut it up so that they can go out now. So this is just again the repeating of the length. And so again, we have these therapy, right? So we have inhibition. We can inhibit RNA polymerase. We can look at antisense and nucleotides that will bind to these cells, that will allow these, that will free up these proteins to go out. So now that we know that this is, so once we know it's an RNA-based thing, it's possible that we can design RNA molecules to come in and free these proteins to go out. So that's the end of it for me. Several diseases have non-traditional genetic mechanisms. So the idea is that if you understand the epigenetics, if you understand non-coding RNAs, if you understand that all these affect differentiation, you're not surprised that mutations in the 3' or the 5' RTR or even the introns would have caused these devastating diseases. But the good news is that as we understand epigenetics, that we're developing therapies to treat these diseases at the epigenetic level. So you really do need to understand, eventually for your practices, if not for the boards next year, you do need to understand that epigenetics are coming your way and that we're developing targets to treat them. Okay. All right, I'm done with this lecture. Thank you very much. Sorry, I guess, any questions? You're done with the lecture too. Okay.